Something big is happening now. Yes, right now. As you listen to this episode, a small team of scientists focused on the vaginal microbiome and preterm birth in London and the Netherlands are beginning recruitment for one of the most important women's health clinical trials of the decade. A study that aims to see, for the first time ever, whether a vaginal supplement containing a beneficial bacteria can reduce risk of preterm birth. Because the composition of the vaginal microbiome plays an outsized role in preterm birth risk, this trial is being watched closely, and the results are highly anticipated. If the trial shows that women given the probiotic have lower rates of preterm birth compared to the placebo group, the news will not only dramatically energize the field of preterm birth research, but likely result sometime in the near future in drastic changes to the clinical management of preterm birth. And so, knowing how important the trial is, the scientists behind the effort are making sure every detail is managed with as much precision as possible. Right now, they're sifting through the medical records of newly pregnant women, identifying those at high risk of preterm birth, checking on the refrigerated stocks of the probiotic bacteria at the center of the trial, and running last-minute tests on the technology that will assess the results. The trial, referred to as FLIP2, is led by Dr. Phillip Bennett, a co-director of the March of Dimes Prematurity Research Center at Imperial College London, one of the leading research groups on preterm birth and the vaginal microbiome in the world. Expected to last about 2 years, FLIP2 represents the gold standard of scientific research, a randomized controlled trial, or RCT, that will compare the preterm birth rates of two groups of women, those who receive the vaginal supplement and those who receive a placebo, providing the most reliable evidence of the efficacy of a bacterial therapeutic to reduce preterm birth risk. This trial is unprecedented, and we've got the man behind the effort right here. This episode is about the historic FLIP2 RCT. Don't go anywhere.
The views expressed on this podcast may not represent those of March of Dimes or its staff. Information presented on this podcast is not medical advice and should not be construed as such. When in doubt, seek competent medical advice. Now, on with the show.
I'm here with Dr. Phillip Bennett, who's professor of obstetrics and gynecology and head of the Department of Metabolism, Digestion, and Reproduction at Imperial College London. He also is one of the co-directors of the March of Dimes PRC. Dr. Bennett joins me from London. Phil, thanks for being here.
Very welcome. Hello.
Congratulations to you and the team for getting here. Being at the doorstep of a big RCT did not happen overnight, and there's been a lot of hard work that preceded this. I want you to explain, if you can, a little bit about why my introduction was so optimistic. This isn't just hopeful thinking about this probiotic, is it? Because we already know that the bacteria these women will take, the bacteria in this probiotic, is called Lactobacillus crispatus. It's been shown to be protective against inflammation, which can cause all sorts of problems for women's reproductive health, from BV to HPV to preterm birth. It's kind of the darling of the vaginal microbiome bacteria at this moment. There's a lot of high hopes pitted on this bacteria. Your group and others did a lot of work in this field, setting the stage for this study. You and your two co-directors of the PRC, Dr. Lynn Sykes and Dr. David McIntyre, you guys have meticulously pieced together many of the pieces of this puzzle over the last 5 years. Lynn, for her part, has shed light on the biological pathways of vaginal microbiome-driven preterm birth, illuminating a complicated domino effect of signals that begin with inflammation and end with the onset of labor. And David has spearheaded the creation of a test that can identify in under 2 minutes the types of bacteria inside a woman's vaginal microbiome and say whether those bacteria are causing inflammation. And you have, in the predecessor of this trial called FLIP1, shown that it's possible and safe to colonize the vagina with the good Crispatus bacteria via a vaginal supplement, the very vaginal supplement we're talking about today, that we absolutely do hope will lower preterm birth risk in these women. So there's been a lot of buildup to this moment, hasn't there?
Yes. Yeah, absolutely. Yes, a lot of build-up, a lot of work that we've done over the past 5 or rather more than 5 years. Quite a lot of work being done in the other March of Dimes PRCs, but also a lot of work done by other people right across the globe. So I think we now recognize that what we would call host microbe interactions. In other words, the mum mounting an adverse immune response to the bacteria living in the vagina is one of the key drivers of particularly early preterm birth. And the problem that we have is that the treatments we currently try to prevent preterm birth, which is progesterone, for example, or putting a stitch in the cervix, those things don't impact this mechanism for preterm birth. So if we can now find a way of treating this problem and preventing it, we could make a big impact on the problem of prematurity. So your introduction is not entirely false and unhopeful, if you see what I mean.
Absolutely. And maybe you can help me understand how big of out of all preterm birth that happens, spontaneous preterm birth that happens, how much of it is driven by the vaginal microbiome? I asked David McIntyre this question, and we kind of concluded, maybe 40% would be accurate. Would you agree with that, or is it higher, lower than that?
No, I think that's probably about right. It's less than half of the cases of early preterm birth, which you could say is before 34 weeks. When we go beyond 34 weeks, there are lots of other causes of preterm birth, which probably are not linked to the vaginal microbiome. So the proportion that it would affect above 34 weeks, not so much. But before 34 weeks, I'd probably around about 40%, I would agree. That's about the right number.
Yeah. So that's, almost half of every baby born before 34 weeks will be born as a result of vaginal microbiome during preterm birth. So, wow, the bacteria inside the woman's microbiome is extremely important. Let's talk about the title. It's called Flip2, catchy title. Why is it called Flip?
Well, it's only called Flip because when we first started the first study, which was Flip 1, we thought, what are we going to call it? You have to have an acronym, and we want to change the microbiome. We want to flip it from an unhealthy microbiome to a healthy microbiome. So we thought, okay, we'll call it Flip. Most studies, the first study is called Flip and the second one is called Flip 2. Well, we really hoped there was going to be a Flip 2, so we put our money where our mouth was and called it Flip 1 in the knowledge that there would be a Flip 2. And indeed, now there is going to be a Flip 2.
Yeah, and brilliant transition to my next question. You just mentioned Flip 1 had the desired outcome, which is that this bacteria was able to colonize the vagina. So tell me a little bit about Flip 1, what you were hoping to see, and what in fact happened.
Yeah, so what we didn't know was whether or not, if we put this bacterium in to women in pregnancy, whether or not it would actually grow and establish itself there and colonize the vagina. It was entirely possible that we could put it there and it would just die and wouldn't take hold, if you like. So the study was set up for two reasons. First of all, to find out if the treatment was acceptable to people to take during pregnancy. And we found that it was very acceptable and didn't seem to be associated with any adverse problems. Nobody really didn't like taking it. And
when you say acceptable, you mean safe, right?
But both safe and acceptable.
Okay.
This is obviously something which needs to go in the vagina. It's introduced using a little plastic applicator that introduces the bacteria. They're in a sort of little pill, if you like, that ends up in the top of the vagina. So it's a little bit invasive. And so the question is, would people feel comfortable doing that? And the answer was yes, they were comfortable doing it. But yes, you're also right. We were looking for safety. So we wanted to make sure there were no adverse consequences of giving this bacterium. We wouldn't have expected any adverse consequences because it's a natural bacterium that would naturally grow healthily in the vagina. So we were pleased to find that didn't happen. And then the other and the main reason for doing the study was to see whether the bacteria would actually survive and colonize the vagina and replace the bad bacteria. And I'm pleased to say that it did do that very well in the great majority of people. Not absolutely everybody. And we're working on trying to understand why the very few people who didn't colonize. But most people did. And importantly, we stopped treatment about halfway through the pregnancy. But in almost every case, the bacteria continued to grow normally right throughout to the end of the pregnancy. So it pretty much did everything we wanted it to do.
Well, fantastic. You mentioned the word acceptable. I'm guessing that you were giving this to women only during pregnancy, or did they take it before they got pregnant? How many times was this suppository inserted, and at what stages of pregnancy?
So they take it in from early pregnancy onwards. They started at 12 weeks.
Okay.
And the course of treatment involves 5 one-day treatments, so one treatment a day for 5 days. And then in this study, we then gave it every week for another 6 weeks. So it begins fairly intensively, and then it spreads out on a weekly basis once you go beyond that.
Okay. So a lot of bacteria here inserted into the vagina, right? I don't know exactly how much is in each suppository, but a fair amount.
Yes. Well, enough, because we wanted more than just to get some bacteria to grow there. We really wanted it to be the dominant bacterium and to effectively prevent all the other bacteria from growing. And that's what it did. So once we had given a few doses, in the majority of cases, the great majority of bacteria there, 99% of the bacteria present, were the bacteria that we put in. So it had sort of kicked out all the bad guys, if you see what I mean.
Yeah, absolutely. And this was a group of high-risk women?
Yes, this was a group of ladies who were already at high risk of preterm birth, mostly because they'd already had a preterm baby, and therefore, of course, they would be highly motivated to be in a study like this.
Yeah, absolutely. And you mentioned that that one of the reasons that you put in so much bacteria is because you wanted them to kick out the bad guys. When Lynn Sykes was on the show, she familiarized me with this term: the good, the bad, and the ugly. So we're talking about the different types of bacteria inside a woman's vaginal microbiome: the good, which is the Lactobacillus crispatus; the bad, and the very bad, or the ugly. But so those are 3, but there's actually 5 community state types, right? Can you talk to me about, or are there, and can you talk to me about how those changed in the women in FLIP1?
Sure. Well, the 5 community state types, essentially, there are 4 different lactobacilli that are commonly found. And then if you have no lactobacilli at all, then you have often a lot of bacteria that are very bad. So that's what you would call the very bad. And the good is Lactobacillus crispatus, which is the lactobacillus that we're going with. And that's good because it's very good at excluding other bacteria, and it has anti-inflammatory properties. It makes acid. That creates a healthy environment that kills off other bacteria. The bad is the one lactobacillus which doesn't have these good characteristics. It's called Lactobacillus inners. It's a slightly smaller bacterium, and it doesn't do the good stuff that crispatus does. Now, there are 2 other lactobacilli, Gasseri and Jensenii, but we don't see those very commonly. So they are so-called community state types 2 and 5, but we see those very infrequently. So really it just boils down to crispatus, good, inners, bad, no lactobacilli, or we call dysbiosis, which is the ugly, as it were. Now, in the study, about half of the women had what we would call a good microbiome to start with, and the other half had either bad or ugly. And by the end of the study, more than 95% of women now had only the good bacteria. So pretty much whenever there was either bad or ugly, our good bacterium was able to displace it. But as I mentioned earlier on, there were a couple of people where that didn't happen. There was one person where it didn't displace at all, and there's one person where once we stopped treatment, something else grew back. So it's not perfect, but it's pretty good. In fact, it's better than we hoped it would be, so we expected it to be.
Phil, you said that some, or maybe half of the women in the study to begin with had good bacteria. So that is Lactobacillus crispatus. Silly question. Why are they at risk of preterm birth? I mean, is it because it's a different phenotype for them?
Yes. I mean, it's not a silly question. You're absolutely right. So again, not all preterm births before 34 weeks are due to the bacteria. There are other reasons. And for sure, we do have women who deliver preterm who have a Lactobacillus crispatus dominated microbiome. So that is probably because the mechanism of preterm birth in their case is something completely different, which is not being addressed by this treatment. It's just possible that not all crispatus are the best, if you see what I mean.
Yes,
And
David and Alexia and DeCoon
yeah,
are doing that research. Yeah,
it might be that if you replaced a not so good Crispatus with a better one, you might do better. But that's somewhat for the future.
Okay, very good. So continuing our discussion about Crispatus, the Crispatus supplement that was used in this study in Flip 1 and in Flip 2 essentially is branded as Lactin V. This is the name of this supplement, right? It contains a Crispatus strain called CTV05. My first question about this CTV05, do we know which of the Crispatus strains is dominant in there? Is it only one strain? Is it a mix of multiple strains of Crispatus?
So in the treatment, in the live biotherapeutic, as we call it, the actual treatment, it's one single strain. CTV05 is a single strain.
A single strain, okay.
Organism, so monoclonal, if you like. It's interesting to ask the question, what do you find in women? Because that is a bit of a mixture. Some women have a single strain. Some women have a mixture of strains. And there's quite a bit of work going on at the moment to understand whether a single strain is good or a mixture of strains is good, and that's work that's ongoing. But CTOV05, in our current state of knowledge, does all the good things that we want a Lactobacillus crispatus to do. It makes lactic acid, it makes antibiotic-like compounds which kill off other bacteria. It has anti-inflammatory properties. It's covered in a cell surface protein, which the shell,
shield,
exactly, which has anti-inflammatory properties. Wonderful. So in our current state of knowledge, it's a good candidate.
Mm-hmm. And I want to say to any woman that is listening, unfortunately, this is not commercially available right now. It may be in the next 5 years or something like that, but it is currently not available. There is no Lactobacillus crispatus strain that has been FDA approved, I believe, in any country in the world. But this is something that I think a lot of women who are scientifically kind of inclined and care about their vaginal microbiome are anticipating. So currently not commercially available, in the process of becoming potentially. And like Phil said, a lot of work happening on ongoing about what is the best strain and whether it's a mixture or a single strain. Now, Phil, a question about Lactin V. Is Flip 2 the first RCT that is using Lactin V or any live bacteria for the reduction of preterm birth?
So the first thing to say is it's not the first RCT using Lactin V, but it's the first RCT in pregnancy. So Lactin V has been used in a non-pregnant patient population to see if it will reduce the recurrence of bacterial vaginosis, which is a condition that you can get if you have those bad bacteria in the vagina. And it was actually because of that study that we got the idea that it might be a good thing to use in pregnancy. And actually, we get better colonization in pregnancy than was got in the non-pregnant trial.
Fantastic.
And that's probably because the hormones in pregnancy are very good at encouraging the growth of lactobacilli. So that's why we do very well in terms of getting the bacterium to grow. And in terms of, is it the first trial? It's not the first trial of probiotics or even probiotics containing lactobacilli to try to prevent preterm birth, but all the previous trials have administered the treatment orally. And there isn't evidence that anything you take orally will end up in the vagina.
Of course, yes, of course.
So there's people who've tried to deliver live biotherapeutics orally, but it doesn't work that way. So
but not for pregnant women, right? Not for pregnant women.
No, there have been trials
for
in pregnancy, but only oral probiotics. And at the present time, the evidence is that it doesn't work. There are still some trials ongoing. I don't want to prejudice the results of those studies. I actually think that the answer is going to be vaginal live biotherapeutics, as we are doing in Flip 2.
Yeah, absolutely. Okay, now this is a question. So you guys are the first ones to do it vaginally with pregnant women using Lactin V. Was there any other group that around the same time as yours also wanted to do the same RCT? I'm essentially asking the question of whether you got these exclusive rights for this RCT because you guys are such a renowned group, research group, or were there any other competitors, shall we say, that were trying to do the same thing at the same time, yet you got the rights?
Yes, I think that we probably identified Lactobacillus crispatus as being an important protective bacteria in preterm labour. We were the first group to show that back in a paper in 2016. And we almost immediately got the idea, well, let's see if we can find Lactobacillus crispatus that we can use clinically. As it happens, we happened to come across the company who make this live biotherapeutic at a conference. And, that's the great thing about conferences. You may go to see the presentations, on the screen, but often it's what you do in the back rooms and have at coffee time that's most helpful. So I guess we just got in first because we thought of the idea first. And then once we had conducted the Phillip 1 study, which incidentally, although it wasn't designed as a randomized controlled trial, so it wasn't intended to show that it could reduce the risk of preterm birth, nevertheless, it did reduce the risk of preterm birth in that group of patients. That's only a very small number of patients, so it doesn't really prove it, but it was enough for us to get, intellectual property patent protection on that live biotherapeutic for that indication, hence why we're now pursuing it, in the next trial.
Fantastic. And what would you expect the preterm birth rate to be in the Phillip 1 participants without the therapeutic, and what was it actually?
So we would have expected it to be around about 25-28%.
Okay.
And it was about 12%.
Wonderful.
So that's much higher than in the background population, but you'll appreciate this is a group of people who are already at high risk.
Yeah, wonderful. And just a random question: are you giving the women more crispatus in Phillip 2 than you would have given them in Phillip 1, or is it the same amount?
Well, we're not giving them more with each dose, if you like, but this time we're continuing the dosing out to 34 weeks.
Okay.
So the reason that we stopped at 20 weeks in Phillip 1 is because we wanted to know whether we would get permanent colonization. So we had to stop so we could see if it went out. In most people in Phillip 1, the colonization did push right out to the end of the pregnancy, but in a few people it didn't. While we were giving it, it stayed colonized, and it was only after we stopped. So we thought, well, now in Phillip 2, to maximize our chance that we have a beneficial effect, we will push treatment out to 34 weeks, but not beyond 34 weeks.
Wonderful. Okay, so then let's talk about Phillip 2. So there are 167 women in the placebo group. Does the placebo group get something? Gets like a suppository with nothing in it?
Yes, exactly. Both they and the midwives and the doctors looking after them will not know whether what they are given is the active thing or the placebo. So it's a double-blind randomized controlled trial. Nobody will know until the end of the trial who got the live biotherapeutic and who got the dummy, if you see what I mean. And that, as you said, is the gold standard of a randomized controlled trial.
So 167 in the placebo group and 167 in the treatment group. Most of these women will be recruited in London, but there will be a small group recruited from Amsterdam. What is this connection to the Netherlands?
Well, so we would like to show that this works in a number of different patient populations. So in London, we have there's quite a lot of ethnic diversity in our patient populations in London. And it's only a small number of people, but at the moment we seem to have the impression it works in everybody of all ethnicities. So we've gone out to Amsterdam for two reasons, really. First of all, to broaden the populations that we're looking at. And secondly, because we would like this to be a treatment that we can use in the UK and in Europe. And therefore, European regulatory agencies will say, well, let's have a look and see if it works in a European population. So you might ask the question, why don't we do the trial in the United States?
That's what I'm thinking. There's a lot of people here who would like to be able to have regulatory approval.
Yes, well, the answer is that when we approached the FDA to do the study, they were very slightly nervous about the level of proof that this is safe in pregnancy. The British equivalent of the FDA, the MRHI, and the European equivalent, the EMA, They were both happy with the data that we had and said, yes, it looks safe. The FDA said, no, this is pregnancy. We really want to make it absolutely safe. So we are going to include the safety information from the FLIP2 study when we go back to the FDA to ask them, then now can we do a trial in the United States?
So is there going to be a FLIP 2.5 USA version?
FLIP 3, yes.
FLIP 3.
Yes.
Okay, but we'll have to wait a couple of years for that, right?
I think so, yes.
And if the Americans were already included in FLIP 2, would you still require a third trial to validate? Or is this just to include the American population? Or do you actually need another level of validation here for a FLIP 3? Is that necessary?
Generally speaking, most regulatory authorities want to see 2 independent trials that prove...
So you will need the 3.
So there always needs to be 2. Well, the FLIP 1 is not a randomized controlled trial, so they wouldn't really recognize that. There's too much chance that there could be other reasons why it seemed to work. So we need to do at least 2. And usually an individual nation's regulatory authority wants to see at least 1 trial in their own country. Mm-hmm. So in the USA, for example, the FDA will want to see at least 1 USA study. So hopefully we'll have 1 European study, FLIP 2, and then later we'll have 1 in USA study, FLIP 3, and then we can use it clinically.
But then you'll need 1 in Australia, 1 in Canada, 1 in Africa.
Not all of the nations require a trial in their own country.
Okay.
So the countries that you mentioned would probably accept USA and UK data as applicable to them. But also, once you introduce a treatment, you can do so-called post-marketing studies where you basically audit what's going on in those countries and you look to see whether or not you're getting the expected effects. So you don't stop monitoring what happens at the point when you release the product. You would carry on looking.
Yeah, well, speaking of carrying on looking, you're going to check in on the babies who were born to the women who took the supplement. So the study will be complete in 2 years. The women will obviously have given birth already, but you're going to, and at that point, at 2 years, or 2 years after the birth of the babies, you're going to check on these babies. Why check on the babies? Yeah,
so that's going to be a separate outcome, as it were. The principal outcome of the study is, do people deliver preterm or not? And we'll know that soon after the study is finished. But to some extent, the benefit of preventing preterm births is to make the babies better. We know that babies born before 34 weeks, and especially those born before 28 weeks, are at high risk of various disabilities as they grow up. So we want to know that we have a lower rate of those disabilities, or we'd like to find a lower rate of those disabilities in the babies that were in the group that had the live biotherapeutic. So we will follow up the babies at 2 years of age, which is a good point in their lives to work out whether they're developing normally or not.
Yeah, and what you're saying is even if these women did deliver after 37 weeks, for example, which is the cutoff of preterm birth, we want to make sure that their babies are showing that term development as well, right?
Exactly.
Because sometimes if you're born at 40 weeks, you could still have a baby that is developmentally premature, or you can have a baby born at 37 weeks or maybe even 36 weeks that is looking like a full-term baby. So that's what you mean there, right?
Well, it is, but also you could imagine that there are some situations where the baby would be better off being out of the womb than in the womb. Let's imagine, for example, if the mother has an infection, and that could harm the baby in the womb. So actually, the baby's best off out of the womb. Now, if we have a treatment that stops that baby being born, it's just possible it could make the situation worse. So the baby is born later, but actually develops disabilities that it might not have had it been born earlier. Now, I don't think that's going to apply in this trial, but we have to make sure that it doesn't apply, and that's why we follow up the babies at 2 years.
Yeah, and just a question about infection-related preterm birth. Is it very obvious that the mother has an infection, or is it possible to miss that the mother has an infection and the birth happens and the baby is impacted? Or is always a million red flags? You're going to either give antibiotics, or as we know, especially in the case of bacteria, not good. Antibiotics could trigger another phenotype of preterm birth. But are there lots of red flags with infection, or is it easy to miss?
No, unfortunately, many cases where if you like, bad bacteria get into the uterus and might then start the processes that lead to preterm labor. In many of those cases, there isn't any signal.
Wow.
The mum isn't unwell, and you do blood tests, peripheral blood tests, and you don't see any changes until things get to be really quite bad. So there is a link between the condition that we're trying to prevent, which is host microbe linked preterm birth, and infection. Because essentially what happens is if you mount an adverse response to the bacteria, it causes the cervix to soften and shorten and open. And then bacteria that are good at climbing up through the genital tract can climb up into the uterus and set up shop in the uterus and can cause an infection. So we think that we will reduce the risk of that happening in this study, but we have to prove it.
Tell me why, again, you think that it's going to reduce? Is it because the cervix is going to be more closed?
Yes.
As a result of the good bacteria being in there?
Exactly. So the link between the bad bacteria and preterm birth, there's an intermediate to that, which is that we know that we can predict that in most cases by seeing the cervix shorten and eventually open up. But this happens later in the pregnancy, and possibly when it's too late for us to do anything about it. So one of our secondary outcomes in this study is whether or not at around about 18 to 22 weeks, the mother has a short or a normal length cervix. So that's something else that we're going to be looking at.
Interesting. I had no idea that the bad and probably the ugly also have another terrible outcome, which is that they open the cervix. Not only do they cause the immune response, but they open the cervix and allow infection.
Well, the immune response is what drives the cervix changing.
Oh, of course, because they're preparing for birth. Yes,
exactly.
Interesting. Okay. Yeah. Very interesting. When can we expect to see the findings of FLIP2? What year? 2028 or earlier?
Yes, I think probably that's right. I mean, you've said 2 years, but it takes us a little while to get the trial started, and it will take a little while afterwards to do the analysis. So I think, probably 3 years from now is a good estimate.
Okay, and what will be the next step if and when the RCT shows lower rates of preterm birth as a result of this suppository?
So, well, I think that the next step at the present time, two things. The first question will be, who should we give this treatment to? In the FLIP2 study, we are giving it to everybody who is at risk of preterm birth because they'd had a previous preterm birth. We're not testing their microbiome and then only treating them if they have a bad microbiome. We're treating everybody. But it might be that it's better to test it and then only treat the people who have the abnormal test. And you mentioned that my colleague David McIntyre is devising a test which can tell you about the microbiome quite quickly. So we're going to be doing those tests in FLIP2, but we're not going to be telling the doctors the results of the tests. So one of the things we'll find out after FLIP2 is not only does lactin V reduce the risk of preterm birth, but also is it better to treat everybody, or is it better to only treat the people where we see there's an abnormal bacterium present? And we'll have the answer to that. And that will then help us to design the next study, maybe the FLIP3 study, which maybe we'll do in the United States of America, which will either be a test and treat study. So you come, say, at 12 weeks in your pregnancy, you have a test. If your bacteria are fine, then that's fine. We don't do anything. If your bacteria are not great, then we treat you. Or it may be a treat all comers, but it'll probably be a test and treat. And the reason for that, of course, is at the moment, we're taking people who are at high risk. They've already had a preterm baby. But we want this treatment to be available to anybody, even those who are apparently at low risk, even those who are having their first baby. So we don't want to treat the whole obstetric population, thousands and thousands of women. So we ideally would have a test, and then we would tell you whether you need the treatment.
Yeah, absolutely. I think certainly I speak for myself. I would love to know, I've said this multiple times, I would love to know who's inside, and what risk they carry. So especially because every woman reacts differently to these bacteria, right? That's why we know that some of these numbers don't make sense. And it's because we all have an individual kind of immune response. So I think that's fantastic.
That's a hugely important point, actually, that not everybody with the bad bacteria will have a preterm birth. Most
Right.
people with bad bacteria don't have a preterm birth. And exactly as you say, it's a combination of the bacteria and the mum's immune response to it. Luckily, the test that David McIntyre is developing can tell you about both the bacteria and about the immune response. So, we may be able to really carefully pick out who is likely to benefit from the treatment.
You guys are the dream team, aren't you? You, David, and Lynn. And I know that there's a whole army of other unbelievable people at Imperial and now at Robinson, where David is, and certainly around the world that are not affiliated with you guys. But, Phil, we're talking here about Lactin V for preterm birth, the DESI, which is going to check the inflammation, and the community state types. These are really, really translational milestones that you guys are currently working on. I mean, it's an exciting time, is it not?
It is an exciting time, and I have to say it's March of Dimes funding that has enabled us to push forward these things very, very quickly. Without that funding, and particularly if we needed to apply every couple of years for a new grant to push things forward, it would have gone much more slowly than it actually has. So it's been a very exciting time for us since we joined the March of Dimes PRC family back in 2017.
And is this Flip 2 the most exciting, or one of the most exciting trials of your career?
Yes, it is. I haven't done very many clinical trials. I'm not really a clinical trialist. I'm more of a scientist. Actually, the few clinical trials that I've done personally haven't shown particularly spectacular results. This, however, is particularly exciting to me because I began my research career back in the early 1980s discovering that lactobacilli are anti-inflammatory and the other organisms are inflammatory. And so here I am 40 years later, actually being able to find a way to reverse that situation using a therapy. So for me, there's a sort of
coming full circle.
Exactly, coming full circle in my career.
Were you one of the first or the first to identify that Crispatus is good?
Well,
anti-inflammatory.
I was a very young researcher as part of a team at Hammersmith Hospital in those days. So I wouldn't claim all the credit for myself, but yes, we were the first group to show that. Yes.
Wow. Well, this is very, very special. Now we talked about the paper probably coming out in 2028. How exciting this is! We're on. So does it ever get easier, Phil, to wait 3 years for the results of a trial? You've been doing this for, like you said, you're not really a clinical trial guy, but obviously you've done a couple of them. But it must be very difficult to have to wait so long to see what are the results. I mean, you at least have to wait 9 months, right?
Yeah. No,
so maybe
true.
It's not 3 years. Maybe it's just 9 months.
No. Well, it's absolutely true. I mean, we do have to wait a long time for the results of the trial, and that's it. It's not. These are not experiments that are being done in little fruit flies. Where you can get the result in a few days. It's worth waiting for, I think, is probably the thing I would say about it.
And how will it reshape the maternal health crisis in America, do you think, and to a smaller extent in the UK, where we know that there are also very high preterm birth rates? What does this have the potential and the capacity to do and to change? I mean, in your lifetime, something that you've been a part of for 40 years.
So I do genuinely think that what we call adverse host microbe interactions are a very important driver of early preterm birth, especially in the United States of America and in some of the populations that we see in the UK. And as I said before, at the present time, the treatments that we use to try to prevent preterm birth do not make this situation better. They just don't touch it. So we have a mechanism for preterm birth which is an important driver of preterm birth, which at the moment we have no treatment for. If we were able to develop a treatment for this, I think it could seriously impact the risks of preterm birth, particularly earlier preterm birth, and could be revolutionary. Certainly that's what I would hope it would be.
Well, that makes 2 of us, and I'm sure many more. A very exciting time for you and for your colleagues and for the entire field, the preterm birth field and the bacterial field. And Phil, thank you for being here. I hope you'll be back to tell us about the results of the trial and anything else that you're working on in the meantime.
I absolutely look forward to it. Thanks very much.
Yeah, it's been an absolute pleasure.
Okay, cheerio.